Well, good morning and good afternoon to everyone who has joined us. Today, our virtual event features Pedro Mendez. He is a senior scientist in our Research Applications and Collaborations group here at Mission Bio. And today he's going to give a great overview around why single cell biology is so important. And so without further ado, I'll turn it over to Pedro.
All right. Thank you, Hannah. Good morning and good afternoon to everybody. I hope you and your loved ones are safe and healthy. We're going to give a brief overview about how single cell technology can really improve your research. It's expected that 1 out of 3 of us will develop some sort of different tumor, and that basically represents 2.5 billion populations of the whole world. But on the other hand, everybody has 37.2 trillions of cells, and few of them are needed just to acquire somatic mutations or genetic alterations or epigenetic dysregulation to start the transformation into a malignant cell. And so we are going to share some of the data that we have been generating in the lab, but also I wanted also to share a couple of experiments that I did back in 2010. So back then, it was the era after the gefitinib, erlotinib, and the impact of those two tyrosine kinase inhibitors in a small subpopulation of non-small cell lung cancer, in which mutations in the tyrosine kinase domain, it was basically activating constitutively the function of the EGFR. And so they discovered that those drugs, they were super active in those patients with those mutations in the tyrosine kinase domain. In 2010, after that era, the squamous cell carcinoma of the lung, it was still treated with cytotoxic drugs. And there was a need to try to find other oncogenic addictions that can be potentially targeted with other drugs. And I remember that a couple of friends from Germany, they published a potential subpopulation of the squamous cell carcinoma of the lung that they were potentially addicted to fibroblast growth factor receptor 1 gene amplification. And so we quickly moved and we started to analyze a little bit of our samples. And this is a sample, as you can see, this is a H&E staining slide. And so what we did it was to get another section of the same tumor. And as you can see, we use laser capture microdissection to enrich the analysis for the tumor, phenotypically tumor cells. So basically in this experiment, what we did it was to measure the copy number variations. And because we had very limited amount of tissue, we just selected this region down here. And as you can see, I highlighted using the two red solid lines, the range of normal gene copy number. Between the lower solid line and the dashed line, it's considered it would be approximately diploid copy number. Between the dashed line and the upper solid line, it would be considered as a region of the copy number value, which can be associated with a copy gain. And over 2, it would be basically gene amplification. And as you can see in this sample, FGFR1 was slightly amplified, SOX2 2, and PIK3CA was slightly over the limit, while the rest of the oncogenes, they were in diploid values. Because we wanted to confirm the data, basically, we got another region of the same slide, just few micrometers away from the same region. And when reanalyzing that sample, we realized that the FGFR, though those cells, they were phenotypically tumoral, FGFR was diploid, PDGFR alpha and KDR, they were mutated. And of course, that makes sense because they are in the same chromosomal band. SOX2 and PIK3CA, they were also amplified, but in this case, SOX2 was much more amplified than PIK3CA, while AXL, EGFR, and MET, they were diploid again. So basically, it's giving us a hint that how complex the tumors can be in just few micrometers away from one region to the other, how different this can be. And it's providing a clear insight that the tumoral heterogeneity is important. This is another case in the same study in which basically following up to the previous experiment, this patient, we were able to follow up the patient for 10 years along 3 different resections. So this was the resection at the moment of the diagnosis. Again, between the dashed line and the lower solid line would consider diploid. Between the dashed line and the solid, it would be copy gain, and over can be considered gene amplification. And again, in this case, SOX2, it was slightly over-amplified or in the limit between those in this different, in 5 different regions across this tumor. When we got a new sample in remission, after remission in 2007, we clearly can see that there was this time a huge amplification of FGFR1, SOX2, and MET. But which is important, it's basically that you can see that this, that this tumor region here, it was very clear non-amplified in the MET gene, while it was amplified in FGFR1 and slightly in SOX. So there was, again, a new proof that there was intratumoral heterogeneity. And 2 years later, when the patient relapsed again, we got on a small biopsy. We did exactly the same thing. And we realized at this time it was PDGFR alpha that it was picking up, and SOX2 it was just in the limit, and AXL was also slightly gained. So those were just few examples. For a long, long time, doctors and researchers, we've been looking at the phenotype, right, under the microscope and trying to get new insights on how correlate the phenotype with the patient outcomes. Then when the NGAs came in, it was a huge boost because we could really sequence a lot of samples and very deeply and have a much better understanding what's the genetic makeup of those tumors. But however, we know that bulk sequencing is important, but when we break down at the single cell level, it's when we really see something that we have been waiting for a long time, which is exactly understanding, which is the co-localization of different variants in different cellular subpopulations. And that's providing us with a new tool in which we can really understand what can be the impact of those different coexistence of variants or gene copy numbers or epigenetic or transcriptomics or protein expression associated with it. So basically, when we look at using the platform that we have developed, it's basically we can integrate all those different analytes into a single workflow. So basically, we singulate the cells, we load in the platform, we prepare the libraries in sequence, and then using the software that we are developing, we can analyze the data and very organically integrate, in this case, protein and genotyping data, or CMVs. And so in this way, in a very high throughput, between 5,000 and 10,000 cells, you can clearly integrate organically those two analytes. So now we're going to share just a little bit of some of the data that we have created with some of the collaborators. It's a shame that we cannot share more because it's confidential, and it's most of it is still ready for publication or already submitted for publication, because there are much better examples on why single cell biology is so important, but at least to provide you with a hint on some of the examples that we have been working on. This is a mixture of 4 cell lines in which we mix them at the same proportion, each of them. You can see that it was Raji, K562, TOM1, and KG1. And so in this case, this is a dimensional reduction visualization based on SNVs, on single nucleotide variants. And by using this specific panel, we could resolve 3 populations out of the 4 different cell lines. When we broke it down, this time not just by SMVs, but by CMVs, we could see how the colors correlate to different cells, and we can see how they are starting to migrate in the dimensional reduction, and we can start to see how they separate, but they are not fully separated. When we add on top of it, when we do the analysis just on protein, we can see very similar trend. One population right here, another one right here, another one right here, and another one right here. But it's when we combine difference, in this case, it's gonna be CMV and proteins, that we can start to see how they really separate apart. And when we combine SMV, CMV, and protein, it's when we clearly see all four different populations. And so that's clearly highlighting the power of the multi-omics and how that helps to understand all the subpopulations within a tumor. This is a clinical sample, and this was a collaboration with the Chris Horrigan lab. And so basically this was an AML patient, and when we broke it down again and we visualized the dimensional reduction of all the variants associated with it, just based on single nucleotide variants, we could see that there was a major population of cancer cells and a minor population of normal cells. However, when we added the protein on top of it, and we could see how a minor cluster of cancer cells that before they were hidden in the cancer population, now they are separated because they are CD11B positive. And as we perfectly know, CD11B positive is a marker of poor prognosis in AML. And so the normal, they were separated too. When we break it down by different protein markers or phase markers by UMAP, you guys can see how CD3, it's basically a subpopulation of the normal cells, indicating that this is T cells. The CD11B subpopulation is very clear right here. We have the CD34, which is segregated to the top right part of the UMAP, and the CD19, which is mostly in this region, and CD38, which is completely opposite to CD34 and CD90, and so on and so forth. And when we visualize this in the heat map, we can also see that there was a chromosomal 7 loss. And in addition to the copy loss too. So basically it's a complex sample, but we can resolve all those different alterations in gene and protein expression based on the multi-omics approach that we provide. So summarizing, we basically shown that the study of intratumoral heterogeneity, it really requires a much deeper resolution level, and that's basically at the single cell level. Our platform, it's providing a very organic integration of both genotype and protein expression data. The research of those two, genotype and phenotype, it's going to be extremely important understanding both the tumor biology itself, but most importantly, the interaction of actionable biomarkers and how those are going to impact both the response and the resistance to the therapy. And yeah, that's what we have all for today, and we'll be happy to answer your questions.
Great. Thank you, Pedro.
Yep.
A couple of questions here. I think some people are still maybe not being able to wrap their heads around the fact that you can actually achieve the genotype and phenotype from a single run or even a single cell. So can you just clarify, is it really from a single cell?
Yes. So basically, I guess that they are interested a little bit in the methodology that we apply for it, right?
Yeah, but I think just more generally too, so that it is indeed the actual genotype and protein expression from the same cell and not from a different.
Yeah. Well, yeah, as I said, technically it's very straightforward. So what we do is we stain the cells beforehand, and then we load in the platform. And that allows us to have exactly in the same cell, because the cells, they are encapsulated in our platform and then barcoded. So we have basically in the same cell, cell barcodes that it's tagging both the antibody and the genotypes. And so we can perfectly deconvolute all both analytes from the same single cell. I hope this is helping to understand a little bit more how that process goes.
That's great. So what is the throughput of the platform, and how many cells can we call from one sample?
Yeah, we usually say over 5,000. When you put more analyze on top of it, you can expect between 4,000 to 6,000, 7,000 cells. If you do DNA only, you can go up to 10,000 easy.
Great. So do you observe differences in protein expression from genes in regions of C and V gain or loss?
We haven't done that yet with internal data. We know that we have seen some of those that they are correlated, but this is confidential data that we cannot share at this point. Yes.
Yeah, so in other words, we have seen that, and our platform
Yes.
has been able to demonstrate that. We're not presenting
Yes.
these data sets here, but we do have customers who have shown that, and I think they're prepping that now for.
Yeah, we have examples in which clearly there's a deletion in the region, and we see a decline in the protein expression.
Great. Are you measuring intracellular proteins or cell surface proteins? And how many SNVs, CNVs, and proteins can you measure at once?
Okay, so for the first question, we are working on it. How many? We have been working for DNA targets. I would say we can target up to 500 different amplicons in the same panel very confidently. And for protein, we have been working for up to 25 markers together.
And can you clarify for protein, is it intracellular or cell surface?
So far, it's surface markers. But as I said, we are working on the intracellular too. This is not something that we are able to discuss right here, but yes, we are working on it because we know that it's going to be very important to be able to measure phosphorylated proteins or just expression of the proteins intracellularly. Yes.
Great. And the single cell proteomics, is it a panel or can that also be customized?
That's something probably more the salespeople can answer. I know that we have some fixed panels and also we have some room for customizing, but it's something that probably it will be better answered by the commercial team.
I can add to that, Pedro. So it's right now we are doing custom solutions. We are in early access mode for the DNA plus protein solution, so that can be available. Or it is available today using customized panels. And in the future, we will also have off-the-shelf catalog panels.
Thank you, Hannah.
No problem. A couple more. How many antibodies can be used per sample at the same time?
Yeah, I would say we've been confidently testing 25 different surface markers in the same experiment, and we can see very good quality data right there. And we have done collaborations in which we have basically validated against FACS, against flow cytometry, and the data is really interesting.
Great. And I think you did address some parts of this question, but is it possible to obtain all the omic data just by one single cell? And how many replicate or single cells do you need to have no biased results?
Well, the good thing of the single cell is because the populations usually they are with several cells, most of them. You have a ton of replicates of the cells inside of the same sample. But we have collaborators that they wanted to test that, and they have done 3 and 4 independent replicates for each of those. And so basically, they saw a very good correlation between the populations.
Great. For C and Vs, can you measure both amplifications and deletions?
Yes, yes, you can. We have shown just data on LOH for the chromosome 7 and TET2, but also we've been analyzing data with gene amplification. Yes, and there's a good dynamic range right there.
Great. You showed some laser capture microdissection experiments. Have we been able to replicate that on the Tapestri platform? And would you see even, or expect to see even more refined granularity in the inner tumor heterogeneity?
No, we haven't done that. Those were personal experiments that I did back in 2010, and I hadn't the chance to work on the very same samples while at Mission Bio. But definitely, that those are experiments that are super interesting to do, right? So having the FFPE and the frozen tissue from the same regions and being able to do that, that would be fantastic.
Great. Another question here. How many cells are needed to start with for the Tapestri platform?
Yeah. We are basically loading right now between 100 and 120,000 cells per sample, which is pretty affordable for most of the samples.
Okay, great. Can the platform be used on mouse cells?
Yes, yes, absolutely. We have support for mouse and the pipeline, a specific pipeline for the mouse genome. Yes.
Great. Just a question a little bit on the methodology. Do you want to talk through a little bit, Pedro, just on a little bit of the two-step workflow maybe?
Yes, yes, absolutely. So basically, the workflow starts with a suspension of single cells, right? It's either from hematological samples or you can basically from frozen tissue, you can extract the nuclei. And then basically you encapsulate those cells or nuclei, and we in the encapsulation process, basically what we do is to add the protease. The protease is basically helping us to release the chromatin and have a much more accurate and sensitive detection of the SNVs, minimizing the allele dropout, which characterizes the single cell genomics technology. And so after that, after the protease digestion, what we do is to bring the cells back into the instrument, and we do the cell barcoding, which is basically bringing those encapsulated cells into a new droplet that contains beads that they have the cell barcode. And so those cells, those beads are releasing the cell barcode, and it's basically what it's going to tag both protein and DNA targeted panels. So further down the line in the after sequencing, we can deconvolute and associate every single read from both DNA and protein to the specific barcode from the cell, for each cell.
And then thereafter, the type of sequencing that is required,
Yeah.
with.
Yeah. So after that, we prepare the libraries, and after the libraries, we balance the libraries between protein and DNA to make sure that we give enough sequencing depth for both of them, and then we sequence. It depends. For a regular panel, let's say that we have 200 DNA targets and 15, 20 protein targets. You can basically sequence that in an XSeq mid output with no problem. We've been sequencing for those 3 to 5 samples per MySeq in high output, and so yeah, basically it's pretty convenient, and from the sequencing standpoint, you don't have to over sequence a lot. Yeah.
Great. Yeah, I'll just quickly add that pretty much we're compatible with all of the Illumina sequencers.
Yeah.
Great. A couple other questions, and I can help answer these too. A question around whether we can support rat cells. We've not done this commercially, so we don't have that yet, but we are looking into that. Another similar question is around, are there plans to develop methods of detecting epigenetic alterations such as histone modifications using a single cell technology? Again, this is something that we've heard from our customers and their interest to do that. This is something that we don't yet have the commercial capability for, but it's something that we are considering and can add to our roadmap. Again, just a reminder, if there are any other remaining questions for the last few minutes, please go ahead and enter them into the Q&A box, or feel free to also enter them into the Zoom webinar chat. Okay. Well, without any further questions, Pedro, I want to thank you very much for this great presentation. And again, thank you to everybody who joined us as well. If there are any other remaining questions, we can also answer them if you email us at info@missionbio.com. We can certainly take your questions there. And additionally, this webinar will be available as a video recording if you would like to re-watch the video. You can find it in our YouTube channel for Mission Bio, and we will also be following up with you over email with the appropriate links. Thank you very much for joining, and hope you all stay safe and healthy.